It's hard being a perfectionist. So we're here in London for the Extractables and Leachables conference. You are one of the chairs. Can you tell me a little bit about the history of this conference and why it's so important for the industry?
This is probably the first conference of its type that came into the being, especially in Europe. We started in 2008.
Right.
Probably the first conference after key regulatory guidance came out onto the field of extractables and leachables. And Smithers was first in line to get that conference. We now have a number of speakers from Big Pharma. We have, as an example, GSK here, AstraZeneca here.
Sure.
So we get people from all aspects of industry, from the end user, the pharmaceutical companies, to maybe the suppliers of devices, or even the supplier of materials to those devices. So we have a big spectrum of people that actually come to the conference to learn more about extractables and leachables and why they're important and why people need to test for them.
And why are they so important, would you say?
It's all to do with safety, to maximise patient safety for the container closure system. A lot of the guidance originated from inhalation devices, so the things like asthma inhalers.
Right.
In the past they were used, should we say, dirty materials, so the industry has moved on and learnt in producing ever cleaner materials, but it's all about reducing what could come into the product and what the patient could take. So it's actually trying to maximise patient safety. The current hot topics are for single-use systems, so bioprocessing. So a lot of the medicines are now going to more bespoke, so more biological-based systems that are targeted to much smaller patient groups.
Yeah.
So those manufacturing systems need to be a lot more nimble. So the old plants that were made from stainless steel are difficult to swap and change.
Sure.
A lot of the changes are now used to single-use bags, plastic bags for a better description,
Yeah.
but very high-tech plastic bags. So the biologicals are actually stored in there or passed through tubing that's involved. And the biologicals appear to be more susceptible to what can come out. So we may detect things that are safe toxicologically, but they may affect the biological that then affects the efficacy that is given to the patient. So we're looking at two aspects. What is safe for the patient and how does it affect the efficacy of the drug product? So how well does it perform in the patient? An example could be there are a number of recalls for EpiPens that didn't give the right dose. So if it's something that changes the efficacy of the product.
Can you tell me a little bit about the tools available and how they can be used to, what is essentially to reduce the risk of extractables and leachables?
Yeah, you need a big toolbox. Very volatile species, so we'll use something like headspace gas chromatography.
Yeah.
So we're looking for things that are solvent-free or looking for solvents that could come out. We'll then look for the volatile species, so we'll use gas chromatography, again with mass spectrometry for identification. So that will get us the volatiles, potentially semi-volatile chemical species. We'll then choose liquid chromatography, again with mass spectrometry, but the range of possible ionisation sources. That will give us the semi-volatile to non-volatile. We're then looking at metals as well, so we're looking at maybe ICP, ICP-OAS or ICP-MS. So that will give us the metals, and then there's always the non-volatile residues that doesn't have a chromophore, doesn't ionise. We'll then try and pick that up by infrared.
Right.
So we've covered just about everything. If something slips through, we've given it our all and as best as science at the moment is.
Right.
So we cannot detect everything, but we give it our best shot.
Another sort of hot topic appears to be, the search for unknown contaminants that may appear in a product. Can you tell me a little bit about specific tools that might be useful in those particular areas?
It's typically the more high-powered mass spectrometry. It's having the accurate mass capability to give us more of an idea of what is the building block of that unknown. So the accurate mass will end up with an empirical formula. The MSMS may give us a chance to do what is the breakdown products of that. So how does the jigsaw puzzle of an unknown put together? So we know the overall shape with the molecular weight. How does it fragment? What information can we have around that? Again, there are commercial databases, but those have less impetus or impact on extractables and leachables.
And I've already spoken to a number of people and they mentioned MZ Cloud as a positive for extractables and leachables because it's an open source platform for people to share spectra of various compounds. How does that fit into the new world of extractables and leachables?
It could be a huge benefit. It is fully peer reviewed before data's entered,
Right.
Therefore the quality going in is good. The question is how many compounds that are applicable to ENL are going in. It takes a lot of effort to put the compounds in that database. So it may be pushing what analytical equipment is currently available to see how low can we go of maybe not in a toxicological point of view of what is it safe to consume that tiny amount, but is it safe in terms of an efficacy of the product? So will that leachable that comes out into the product, will it affect the efficacy of that drug product?
Right. That's it. It's as much as you can get in 5 minutes of interview, taken over about an hour through coffee breaks. Brilliant.
Okay.
Right, these guys usually want to do like a bit of a walk and talk footage, just so they can see that we don't hate each other off the camera. I think that, I guess that's the reason. It's like, look, they don't just disappoint.